Today we'll be discussing blood culture-based pathogen detection testing in 5 minutes. To start with some background, let's review a few different pathogen detection tests. The current gold standard to identify bacterial pathogens are culture-based tests. These include Gram staining techniques, mass spectrometry, and other biochemical identification tests. There are also PCR-based or molecular tests, with one example being the SeptiFast test, which can be performed directly on whole blood samples and doesn't require any culturing period. Then there are antibody-based tests, and we're quite familiar with the rapid antigen test for COVID that falls in this category, and there are genomic sequencing tests like Illumina and other next-generation sequencing modalities. And as we said, blood culture-based methods really are the gold standard for detecting and identifying bacteria. But how does this work? Let's say we have a patient that we suspect has a bloodstream infection. The first thing that we need to do is take a sample of that blood. Then the clinical team will start empiric broad-spectrum antibiotics in the hopes that they will fight the infection given the most likely bacterial candidates until we know for sure what bacteria is actually there. So blood is collected in this broth tube, and then that tube is placed into an incubator until we see growth. This typically takes 24 to 48 hours, but the tube may be left in as long as 5 days. If that broth tube becomes positive for growth, we know that some bacteria is present, but we're not sure about the identity of that organism yet, so we need to do further testing. We can plate a little bit on an agar plate and do some Gram staining techniques. And this will tell us whether we're dealing with a gram-positive or a gram-negative bacterium, and if they're cocci or bacilli, and this gives us instant results. We can then plate a little bit more and create an agar subculture, stick it back in the incubator, wait overnight, and do further testing to identify the specific species of bacteria that we're dealing with. And we can either do that through mass spectrometry like MALDI-TOF MS, and this gives us results instantly, or we can do these so-called conventional biochemical identification techniques, which will give us results in the next day. So at this point, we'd know what species of bacteria are causing our patient's bloodstream infection, but we don't yet know what antibiotics are effective against it. So then we can go on to susceptibility testing in another subculture. And we can either do genotypic or molecular testing, which looks specifically for antibiotic-resistant genes or gene products, or we can do phenotypic testing, which exposes bacteria to certain antibiotics, and we just see whether they survive or die off. And we can do conventional testing, which includes a whole host of different tests that roughly takes 24 to 36 hours to give us back results. Or we can do these rapid testing methods, which gives us results within about 8 hours, and this can be either phenotypic or genotypic-based testing. Based on these results, now clinicians know how to tailor down their antibiotics to the specific pathogen identified and narrow that unnecessary coverage of other types of bacteria. Some important terms to note here include time to positivity, which refers to the amount of time between first taking our blood sample and then seeing growth. Then we have time to identification, which refers to the amount of time between taking our sample and then knowing what species we're dealing with. And then we have the time to tailored antibiotic therapy, which is clinically important because we get to tailor and narrow down that broad spectrum coverage to a more narrow spectrum specific coverage. Some of the limitations of blood culture-based testing, really they take a long time to reach the wards, and that leaves patients taking broad-spectrum agents for a prolonged period of time. This delays the amount of time until we can narrow antimicrobial coverage, and this can even lead to changes in the host microbiome. We really don't want to be attacking bacteria that aren't causing our patient any issues. And finally, this increases selective pressure for antibiotic-resistant pathogens. So we definitely want to be more conscientious about our antimicrobial stewardship and de-escalate to narrow spectrum agents as soon as we can. Thank you so much for listening to this intro talk on blood culture-based pathogen detection. In part 2, I will be discussing what randomized controlled trials look like in the world of microbiology, including faster methods of pathogen identification. Thank you.